welcome to the Deep Dive. Today, we're jumping into something really fascinating, electrophysiology in neuroscience.
That's right.
Trying to understand how our brain actually communicates, how it works at that really fundamental electrical level.
Absolutely.
We've got some key questions guiding us, and yeah, the mission today is to pull out the core ideas, what it is, how we study it, and, well, why it matters so much.
Sounds good. Well, at its core, electrophysiology in neuroscience is basically studying the electrical properties of neurons.
The brain cells.
Exactly. The brain cells and the circuits, the networks they form, we're essentially trying to listen in on their electrical conversations.
Listening in. Okay, so how do we actually do that? How do we record those tiny electrical signals?
Well, there are a couple of main approaches. One is called intracellular recording.
Intra meaning inside.
Precisely. You place a very, very fine electrode inside a single neuron.
Wow.
Yeah, it gives you an incredibly detailed look at what that one specific cell is doing electrically. Like isolator one violin in a huge orchestra.
Got it. So that's super focused. What's the other way? Extracellular.
Exactly, extracellular. Here, the electrode is placed near the neuron or group of neurons, but not inside.
Okay.
So you're recording the electrical activity happening just outside the cell. It's maybe less detailed about one single cell, but you can pick up the firing of several nearby neurons.
More like hearing a section of the orchestra from the hallway.
That's a great analogy, yeah. Both methods give us different pieces of the puzzle, really valuable, complementary information.
Now, people often see those caps with wires, right? EEG. How does that fit into listening in?
Ah, right. EEG, electroencephalography. That's quite different. It's non-invasive, so the electrodes just sit on the scalp.
Outside the head?
Correct. And instead of recording single neurons, EEG measures the summed electrical activity of millions, even billions of neurons firing together.
So it's a much bigger picture view.
A much bigger picture. Think of it like measuring the overall electrical hum of an entire city, not eavesdropping on one phone call. It tells us about general brain states like sleep, wakefulness, or detecting large-scale abnormal activity like in seizures.
That brings up something interesting. Brain-computer interfaces, or BCIs. This electrical recording is key there, isn't it?
Absolutely crucial. Electrophysiology is the foundation of most BCIs. By recording those neural signals.
Like the EEG signals, or maybe more direct ones?
Often more direct, yeah, sometimes using implanted electrodes for finer control. But the principle is the same. Record the brain's electrical activity, decode the intention behind it, and use that to control an external device.
Like a prosthetic limb, or a cursor on a screen.
Exactly. For people with paralysis, for instance, it's potentially life-changing technology, direct communication from thought to action.
It's just incredible. So when scientists look at these recordings, especially the more detailed ones, what are they actually seeing? I've heard the term spike train.
Right, a spike train. That refers to the sequence of action potentials. Those are the actual electrical impulses, the spikes, that a single neuron fires over time.
So like dots and dashes in Morse code.
Kind of, yeah. It's the neuron's way of sending discrete messages. The timing, the pattern, the frequency of those spikes, that's the code neurons use to communicate information.
Okay. And you also hear about LFPs, local field potentials. How are they different from spikes?
Good question. LFPs are slower fluctuations in the electrical potential in the space around neurons. They don't represent the firing of a single neuron like a spike does.
So what do they represent?
Well, the thinking is that they reflect the summed activity of a whole population of neurons in a small area, mostly the input signals, the synaptic activity happening between cells, sort of the collective hum or background activity influencing the spikes.
I see. There's another term that sounds quite technical. Patch clamp. What's that about?
Patch clamp.
Yeah.
That's a really powerful high-resolution version of intracellular recording, essentially.
Even more detailed.
Oh, yeah. It allows researchers to isolate a tiny patch of the neuron's membrane, sometimes containing just one or a few ion channels.
Ion channels. Those are the gates controlling the electricity.
Exactly, the tiny molecular gates that let charged ions flow in and out, creating the electrical currents. Patch clamp lets us study how those individual channels work. It's fundamental for understanding the absolute basics of neuronal electricity.
And understanding those basics.
Mm-hmm.
That must help with Diagnosing problems, right? Like epilepsy.
Definitely. Epilepsy is a prime example. It's characterized by abnormal, synchronized electrical activity in the brain.
Which you could see with EEG.
Precisely. EEG recordings are often critical for diagnosing epilepsy. They can show those characteristic spike wave patterns during or even between seizures, helping pinpoint where the seizures might be starting and guiding treatment.
It all sounds incredibly powerful. Are there any big limitations with these techniques right now?
Oh, absolutely. Despite the power, there are limitations. With EEG, for example, you get great timing information, but it's hard to know exactly where deep inside the brain a signal is coming from. Spatial resolution isn't great.
Right.
And even with implanted electrodes, recording clearly from, thousands or millions of individual neurons simultaneously for long periods is still a huge challenge. There's ongoing work, lots of development trying to push those boundaries.
So, okay, wrapping this up a bit, electrophysiology is really fundamental. It lets us listen in on the brain's electrical language, from single neuron spikes
Mm-hmm.
to the big picture waves on an EEG. And this understanding is key, not just for basic science, but for real-world applications like BCIs and diagnosing conditions like epilepsy.
Exactly. It bridges that gap between the brain's microscopic electrical world and our thoughts, feelings, and actions, and even potential therapies.
It really makes you think.
It does. And maybe a final thought for you, the listener. As these electrophysiology techniques get even better, letting us read brain signals with more and more fidelity, and maybe even write signals back in, what ethical lines do we need to consider? How do we navigate that future where the boundary between brain and technology might become, well, even more blurred? Something to ponder.